Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 126th seminar, and I call it a Georgia Tech Day. May is Asian American, Native Hawaiian, and Pacific Islander Heritage Month. The United States has many such months, including Black History Month in February and Women's History Month in March. Having such months may be good, but do we have a white or men's heritage month? The answer is no, absolutely. Such heritage months are a clear indication that the United States still does not have equality or equity in terms of race, gender, sexual orientation, religion, etc., let alone equality for international people. I hope we will soon live in a world where we do not have such months because all humans are equal, regardless of their race, gender, sexual orientation, religion, belief, and nationality. I have high hope because we have passionate young researchers and people potentially solve all the problems we currently have. Okay, it is my tremendous honor to introduce very briefly, I'm not sure I can do so today, but I'll try, our pioneer speaker, Professor Corey Wilson. He does not need any introduction because we all know him. He is a professor of chemical and biomolecular engineering at the Georgia Institute of Technology, in short, Georgia Tech, which I visited this month, a couple of weeks ago, for the EBRC Space Roadmap Workshop, UK-US Engineering Biology Meeting, and EBRC Annual Meeting. More importantly, as you already know, there will be another important meeting in Atlanta again, the SEED Conference, the premier synthetic biology conference, where Corey is the conference chair. That means he has been very busy to entertain all of us to attend one of the best synthetic biology conference in the world. I'll assist him as one of the session chair, and then I really thank him for his service to organize this important conference. And I look forward to seeing many of you in Atlanta less than 1 month later. Corey, thanks so much for your amazing service to the community, especially again, including the SEED conference chair role. Very, very heavy job, a lot of responsibility, I know, because I already done a couple of times before as a committee members. But that's the important work and a lot of job. That's pretty much one thing I'm going to say. And then thank you again. The virtual podium is all yours now. Thank you.
Okay. Thank you for the very kind introduction. I'm going to share a couple of slides with you. I have a short window, about 10 minutes to sort of talk to you about what I do and sort of motivate you for the featured speaker, and that's going to be Andy. Everyone can see my slides. Everything's good?
Yes.
Okay, great. And so I'm going to share a short story, or a shorter version of a story, in our journey to develop intelligent chassis cells. And so this is something that we worked on for the past, say, 5-6 years. And what I'm going to do is try to at least discuss how I intersect with Andy, who is very specialized and steeped in protein design. And so he's currently a postdoctoral fellow with David Baker. Prior to that, he was a graduate student at Northwestern with Chad Merkin. And so it's my pleasure to share this time with Andy. And so without going into a lot of detail, we focus on something called convergence research, specifically in the area of biomolecular systems engineering. This is a space that I pioneered. And so in this space, what we attempt to do is to converge biological engineering, chemical engineering, and electrical engineering. And as many of you know, chemical engineering and electrical engineering are established fields. And the idea is that we are hoping that we can elevate biological engineering to a proper engineering discipline. And so the way we hope to achieve that is by utilizing and being inspired by workflows that have been developed in the two established engineering disciplines. So biological engineering is a very large space. Here we're focusing on iterating between protein engineering, genetic engineering, and metabolic engineering. And so this is where Andy and I share an interest in that he's interested in protein design. I've actually worked in protein design as a postdoctoral scholar at Caltech with Steve Mayo. And so I really deeply understand what he's doing at a fundamental level with David Baker. And so with this program and with this focus, we're applying it toward the development of intelligent biological systems. So by our definition, an intelligent biological system has three fundamental tenets. That is, it has the ability to make a decision. We have a coupled memory structure. And then for advanced systems, we can get them to communicate. And so I'm not going to have time to talk about all of this in 10 minutes. And so what I'm going to do is share a slide with you that summarizes some of the recent papers that we published in this space with regard to the three fundamental tenets, principally decision-making, memory, and communication. And so just a brief overview for the intelligent system, as I mentioned, the two tenets that we principally spent the most time developing are decision-making and memory. Okay, and then we did this in the context of what we regarded as the biomolecular systems engineering workflow. And so that means that what you're going to hear is a lot of complementary descriptions of systems in the context of what we normally talk about from the engineering vantage point. And so the technology that we developed for decision-making is transcriptional programming. And so transcriptional programming has a number of unique features. One of the principal features is that we've been able to integrate the biological circuit or compress the biological circuit. And so this is compression or integration in much the same way that we've integrated or compressed the transistor. And so what you see on the right side is this first image is a large transistor from about 1925. And then over time, what we've been able to do is make the transistor smaller and smaller. Now we can fit hundreds of transistors to a relatively small space. And so the advantage of accomplishing this is that it increases our computing capacity while reducing the resources that we need to actually execute an operation or program. So in much the same spirit, when we compress a biological circuit, that's what we also gain. We increase our biocomputing capacity while reducing the resource burden on the cell, which is important in synthetic biology because we have this ever-increasing size and complexity of biological circuits that we need to address. So this talk, or at least a very short talk, is going to focus on the wetware. We also have complementary software. But with that in mind, I'm going to show you how we set things up. This is set up in much the same way that we would address systems in the context of control theory. We have two types of open loop control systems, a single input, single output control system. And then from there, we can systematically construct multiple input, single output control systems. With that said, we also have the ability to do feedback control, et cetera. And then with time permitting, I'm going to show you how we develop this programming language in the context of being Turing complete. All right. So where do we start? And so we started with protein engineering. Specifically, we engineered two classes of synthetic transcription factors, repressors, which are shown in the blue box, and anti-repressors shown in the purple box. If we abstract away the detail, we can describe the repressor as a buffer operation where 0 input corresponds to 0 output and 1 input corresponds to 1 output. And antithetical to that, we can abstract away the detail and describe the anti-repressor as a NOT operation. Here we have 0 input corresponding to 1 output. And then when we add the input, we actually turn the system off. And so these are not common in nature. And so this is perhaps one of the most important features of transcriptional programming. Sometimes you'll hear me contract that in regard, or call it T-Pro. And so over the years, we've been able to create hundreds of synthetic transcription factors that respond to a broad range of different inputs. And with that, we've also been able to construct these systems using modular design in a way that allows us to network transcription factors. And so that networking feature is important. The way that we achieve that networking is through the utilization and deployment of synthetic promoters. And so here, I'm going to iterate from protein engineering to genetic engineering. And so we can direct transcription factors, at least our class of synthetic transcription factors, in much the same way that you would direct current. You can do it in parallel, you can do it in series, or a combination thereof. And so what I'm going to do is I'm going to focus on one particular architecture. This is called the series parallel architecture as our synthetic promoter. And I'm going to walk you through a fundamental design of a circuit. And so here you start by selecting your synthetic promoter. Again, this is the series parallel promoter. And then you select your 2 transcription factors predicated on the response to that synthetic promoter. So in this case, what we do is we select 2 anti-repressors that share a DNA binding function, but they respond to 2 different inputs. And so on the left, we have one responding to a gray input, and then on the right, we have one responding to a purple input. Once the circuit is constructed, what you have is an objective representation of a NOR operation where 0 input corresponds to 1 output. And then in any other case, you only get no output with one or the other signal present. So NOR is special axiomatically. If you can layer NOR operations, you can write any program that you're interested in. And so this is actually the way that we developed the control system for the early Apollo missions. And so it's not necessarily the most efficient way to construct a program. And that's also true in programming in biology. And so this is where transcriptional programming really takes off, in that it enables us to construct these circuits in a very efficient way. And so what do I mean by that? So this is circuit compression illustrated. So on the left side of the screen, what you see is a classic description of a NOR operation generated using something called inversion. And so in inversion, the punchline is, to construct this circuit generally, you would need 5 inducible promoters. And so using our transcriptional programming edifice, what we can do is we can construct that same NOR operation with a single promoter. And so this is a 5-to-1 compression. And so this enables us to save on the resources or minimize the resource burden, because we typically deploy these systems in living cells. And so the punchline here is that this is scalable. And so what I'm showing you are the 2 most complex logical operations that you construct from a Boolean perspective. And so on the left side, what you see is the compressed iteration of the circuit constructed by T-Pro. And on the right, what you see is the circuit constructed using traditional inversion. And what you get in this case is a 16-to-3 compression on the circuit when we use transcriptional programming to construct that circuit. So you can see that there's a significant compression in the circuit, and we see a similar compression in the XNOR gate. So over a number of years, we've been able to construct these systems in a Turing complete fashion. Namely, we've been able to construct these systems, say, for 2 inputs where we represent all 16 Boolean logical operations. We've recently deployed this in living cells, and so, cells that live in the gut. And so this is an illustration that this is a universal programming language that can be deployed in any cell. And so from here, we've set the platform for living therapeutics. And this is also an intersection between Andy and I, and that he's also interested in developing living therapeutics. And so that is about all the time that I have. With the last 30 seconds or so that I have, what I want to do is I want to thank the people who actually did the work. So this is my research lab. And what I'm going to do is I'm going to turn it over to Andy so that he can share his amazing work. And it was a pleasure talking to you.
Absolutely amazing. So you know what? I follow your work because my training with the Chris Voigt is all about genetic circuit. So I'm glad you continue to do so. And that's absolutely amazing, especially the last one with the real potential application in the human body. and we probably want to deliver real product in near future with the genetic circuit. That is absolutely one amazing avenue we can explore. Thank you so much for a wonderful talk. Okay, so and also you are my academic uncle because you are Stephen Myers
Yes, that's right.
alum, right?
That's right.
I realized now actually.
Yeah.
Okay, so now the main speaker of today with a longer introduction. Dr. Shunzhi Wang, or just simply Andy Wang, is currently a postdoc fellow at the Institute for Protein Design at the University of Washington, as Corey already kind of mentioned, working in the lab of Professor Dave Baker. His research primarily focuses on designing programmable self-assemblies using biomolecular building blocks, including computational protein design, colloidal crystal engineering with DNA, and bio-inorganic hybrid systems. I guess, I mean, everyone now probably agree to the point that we need some computational design and using of AI or whatever computational method to design protein and also whatever genetic circuit analysis, etc. And then the Andy is one of the person who focus on those kind of effort. He obtained his PhD degree from Northwestern University under the supervision of Professor Chet Merkin. His notable works include the discovery of electron-like particles in DNA colloidal crystals and the introduction of reinforcement learning method for the top-down design of protein architectures. I guess this is a natural transition from his PhD work to the postdoctoral work in Dave Baker's lab. I believe I met him last year during a conference where I was impressed by his work and presentation, and I immediately realized he's gonna be future leader, one of the future leaders. And thus, I reached out to him to become a rising star speaker today, and I'm really grateful to have him today. Andy, thanks so much for your time today, and please take it away. It's all yours now. Thank you.
Wow, thank you so much, Teso and Corey, for the very kind words. I'm thrilled to be here and then share my work with everyone. And yeah, let me share my screen. Oops, oh my bad.
Yeah, we're talking about Go earlier together.
Yes, let me share. So yeah, I came from a slightly different background, as Corey and Teso already mentioned, from computational protein design, where I did my I'm doing my postdoc with. So, but I think that is a emerging set of tools to kind of innovate the field of synthetic biology through the design of protein building blocks. And here, it's really hard to follow Corey's talk because that's a truly programmable system. I feel a little bit ashamed by calling my system programmable protein assembly, but we all see how we do it. And then, yeah, let's start. So I'd like to motivate my talk given this picture, which is a living snapshot of what's happening inside cells. It's a lot of things going on, not just a bunch of molecules flowing in. We have a lot of higher order structures. For example, these are clathrin cages, these are vesicles, these are microtubules. So these are hierarchical structures that in our living cells that we need them for function, biological functions. These structures form through the self-assembly processes. And without them, living system will be just a tasty soup. So, but the question is, how do we get it from a soup, which maybe 3 billion years ago our common ancestor were, and to these functional biological living system? So some of the key challenges is all of these self-assembly processes come from these non-covalent interactions. They're highly dynamic yet precise, and then they're away from equilibrium. So is it possible for us to design such assemblies and then guide the building blocks to form structures with emergent biological function? That remains to be a grand challenge of our age. So as you can see, there are a lot of these such structures come from protein assemblies. To name a few, there are a bunch of well-defined complexes. For example, these Catherine coat proteins, they're really important for the cargo transportation. And then nuclear pore complexes, they govern these information exchange, and then these mRNA processing, innate immunity, and then these energy transfer. And from a structure perspective, there are certain organelles, for example, these are microtubules or bacteria microcompartment. They play really crucial roles in the living system. But we have been primarily relying on natural system, and then the field has primarily relied on structural biology to observe what's possible to create nature. But can we create something from our own? Although the first initial steps could be relatively simple, but we believe that in long term, we are able to build these more complex machineries. But it is a challenging task. As we all know from a different length scale, the scaling law, where the atomic scale, the chemical bonding, covalent and interactions are governing. And the micro scale at the cellular level, the gene regulation, the central dogma. At nanoscale, you rely on these supramolecular interactions to create these functional assemblies. As we mentioned a little bit before, they're non-covalent, they're dynamic of equilibrium. Ways to think about them rely on these empirical knowledge has been challenging. So we need design principles and computational tools to help us to go through this process. So my talk will be relatively narrow and then won't focus too much on the techniques, but the rationale behind those. So throughout my academic journey, I've been taking a route from, as a trained chemist, from coordination chemistry, where I designed nucleic acid surface ligands. We chose nucleic acid to study first because the complementary wasn't canonical base pairing interaction are highly predictable and it can be used to design things. For example, the DNA technology, where we're able to use DNA to guide the particle self-assembly into defined architectures. But now we can rely on these. A new revolution we are actually experiencing is proteins start to be designed. I remember 3 years ago when Dr. Frances Arnold first gave her note, she was mentioning that proteins, enzymes could not be designed. However, I would say I think the science is trying to move at a very fast pace that we're going to start to question some of these statements just a few years ago. So today I'm going to share 2 works that in this territory where we want to design programmable protein assemblies. So the grand vision of how we do it is we want to shift from the chemical bonding, where the atomic chemical information, atomic bonding that encoded in atoms, where these interactions can be encoded by DNA and then proteins. And then where we can start to identify fundamental design principles based on DNA bonding, and then getting the knowledge I learned from the PhD to push for the protein interactions, which are fully genetically encodable, and then we have a great tools to develop them. So just want to give a very quick overview of the field of protein design, which is I cannot be able to say much, but just saying that it's a reverse problem of protein folding problem, which has been a 50-year thing, has a long history. And then primarily people have been using the biophysical tools to study this problem to kind of see how protein as a string of molecules to assemble. And then recently has been more and more statistical method where experimental data has been consolidated into understanding the structures. So the forward problem is given a one-dimensional sequence to output the three-dimensional structure of a functional protein. And then what I'm interested in is the protein design problem is, can we provide a 3D structures or functions and then grab a one-dimensional sequence to encode that function? So We have a lot of these natural proteins already in there, but why do we need de novo proteins? Well, there are probably three main reasons behind that. One is the human proteome is probably around 20,000 proteins out there, which is a very small fraction of unique proteins on Earth. And then if you think about the possible protein sequence space, it's only a very, very tiny amount of that. So there might be very likely a subset of possible proteins that are better suited for our technological interest. For example, higher soluble or stable or non-immunogenic structures that can fit our need. And also, we can utilize them as a way to augment biological functions. For example, in making the or engineering metabolic systems. So the grand paradigm is if we learn from biology where the natural sequence can encode structures and then they have some evolutionary functions, we can create a de novo design where what we're trying to work on is a set of rational design algorithm where we can identify new sequences and the structure and functions. Some of these possible outcomes will probably range from the public health and precision medicine with respect to vaccines or catalysis or breakdowns plastic, programmable cell-based therapies, molecular machines, and then even think about the implant system where the human-machine interface can be augmented by protein design. So in a typical protein design workflow, we can view, or state of art can be viewed as we create a functional protein backbone without sequence first that satisfies our need, for example, for binding task or stop flu, and then we assign sequence to allow them to stabilize such conformation. And then we use computational filtering strategies, for example, reverse folding or scoring mechanism, and experimental characterize them. So that's our typical workflow. And then before 2021, people have been primarily relying on biophysics. To name a few examples, I mean, Rosetta software is one of the tools people have been using, and then people has been calculating these energy of the system. And after 2021, deep learning started to dominate, and then there's a lot of tools people developed from all over the field, but just to name a few, for example, the Rosetta Fold Diffusion, ProteMPN, that can perform some of these tasks. So today, I'm going to talk to you a little bit about, in order to design protein assemblies, the backbone generation is one of these more challenging tasks. So in the true story I'm going to share a little bit today, I'll focus on how to generate shape complementary building backbones that satisfy the symmetric need, as well as be able to create a scaffold that be functional and then be tested both intracellularly and in vitro. So this is kind of the first work. It'd be design of self-assembling proteins into predefined crystals. So we all know that the primary sequence can fold in a hierarchical way into secondary structure, tertiary structure, and then quaternary structure. So my question is, does the primary sequence also dictate how do they pack, namely how the crystal form? That is potentially a quaternary structure of packing of the protein complexes. That remains to be a question that the field don't relatively have a certain answer for, because the natural proteins typically crystallize in a tedious experimental screening way. Because of their irregular shapes, protein will probably pack in a low symmetry space groups. So in order to design a crystal, just so that the sequence will predictably form into a three-dimensional packing, there are several challenges. One is a packing preference. Why does the proteins want to pack into one arrangement versus the other? And then they could be very similar in energy, or there's interface orthogonality, because there will probably be three coexisting interfaces. If you want to design them, they need to be orthogonal so that they are not interfering with each other. Sort of, it's just, in order for design things to form a crystal, they have to be atomically level precise. Otherwise, they will propagate and make mistakes. So basically, the challenge we addressed over here is, can we design a single protein that makes three accurate protein-protein interface to support their crystallization? Well, apparently we have been able to do that, and then I today I would like to share with you how we conquer this relatively challenging problem. So the way our strategy to address this problem is a divide and conquer problem way. So we want to sequentially decrease the degree of freedom where the protein monomers that can assemble. First, from a monomer level, they were constrained into forming the cyclic oligomer, and then the cyclic oligomer be able to combine into a polyhedra, which will take a slightly weaker interface. And finally, these polyhedra were able to dock in the three-dimensional space into crystals. So we chose a model system to, for example, docking into a diamond lattice, just because diamond lattice are relatively low packing, as you can see. They're very, very hollow, so that if we actually be able to get crystal of the diamond lattice, they're very primarily through this design interaction. So here is how we design the system. We form this very strong, we want to design a very strong oligomeric interface, and then a cage interface that's a relatively weaker and then crystal interface. We can do that in a more traditional way where Rosetta software were invoked, because this project was done when I first joined the lab where deep learning method was not so prevalent. So we take a docking plus design type approach where we use rigid body sampling to kind of look at the protein-protein interface with slide digitation and assign monomers, rotamers, and then these identity of these sequences to stabilize this interaction. And then we can score these interactions based on the Rosetta software, where you can look at these solvent accessible area of this interface, as well as these predicted binding energy. So by looking at these a gradient of these larger surface solvent accessible area, that means more stronger interactions. So we actually be able to get some very delighted to get some successful results. As you can see, we can combine two trimeric proteins into these two-component tetrahedra particles. And then these particles are docked through these orange interface into these the diamond lattice F4132 space group. They form 100 micron size crystals, beautifully octahedron. And then they see this in small angle X-ray scattering just to make sure that there's no mixed phase crystals out there. And then we took a single X-ray crystallography to look at these exact molecular interaction details. So the oligomeric interface spot on, and then these cage interface are also between these design and then the crystal structures almost identical. And then the crystal interface are also as designed. So because these system is more of a model case where the oligomers are natural building blocks, we wonder if we're able to design crystals that start from scratch, where fully de novo. So here is the case in the design number 2, we create two set of building blocks that are completely de novo. There's trimers and dimers. They form another type of tetrahedra. There's a T32. And then still interacting through this trimer interface. We obtained these cryo-EM data of these particles to make sure these interfaces are as designed, so that we have a higher confidence of where they are. And then indeed, we see these octahedral building crystals, and then the sets show that match with design. Interestingly, the X-ray crystallography also closely matched the design model. As you can see, different interfaces are highlighted over here. However, because these crystals are super porous, we are not able to get very high resolution of that to review details about the crystal. We only see the secondary structures that are pretty close to our design crystal. Lastly, we want to say, okay, well, we want to design things other than the diamond lattice. How about a body center cubic? In this case, instead of tetrahedral, we design two sets of building blocks. That's a tetramer and then a trimer that they form the octahedra. This octahedral will pack like a spherical ball through these dimeric, these three interface, and then form this rhombic dodecahedral crystal habit, beautiful crystals, and then very, very sharp small angle X-ray scattering peaks. And then also we got around 3.5 angstrom crystal structures, suggesting our design crystal, all the interface are as designed. So that methodology can be expanded to much beyond what's possible. I've shown over here. However, we didn't have to spend most of the time to characterize them. But we can see that the different docking methodology and followed by redesign were allowed to create different types of crystals, lattices, as well as increasing the component of the things a protein crystal can carry. We also see note that these type of crystals that designed fully de novo occupy some of these most porous structures we can ever see in nature. So these are genetically encodable porous materials, where, as you can see, they're among the top 5 regarding this highest solvent content, as well as we can do a pretty robust crystal re-engineering, such that we can change their the crystal lattice such that they can isotropic expand or utilize them to as a scaffold to template inorganic nanoparticles arrays. More interestingly, these crystals give some pretty robust crystallization behavior that would not expect from typical natural crystals. As you can see, they're thermally stable at 95 for 1 hour. They still survive, where the natural crystals you probably melt in your hand. They can also roset them in cell lysate. And also we can introduce them to 293 mammalian cells, and then they form these single crystals inside of cells. Well, by introducing the crystals into cells, we see that the cells carrying them do not suffer from so much of stress and then cause cell death up to 3 days. And then these cells carrying crystals can divide normally. So this is a collaboration with Ding-Chan Lin's lab. So that opens a lot of opportunity to think about potential, carboxysome type of architectures or cellular factories that we can put enzymes into these crystals and then utilize them to as a mechanism to concentrate molecules for metabolic engineering or system biological approach. So we're pretty excited about this new direction that emerged from this novel type of materials that previously not accessible to be introduced into the synthetic biology realm. So next, I would like to talk a little bit about how we leverage the power of AI in creating protein architectures that are so far been proven very challenging to make. For example, some natural assemblies through evolution, these viruses or these gas vesicles or evolved proteins, you can see when you look at these structures, they're so pretty, and then all the subunits fit perfectly into each other. And then they perform their functions. So that made us amazed. How do we design these proteins that self-assemble into these beautiful architectures? Well, this very hard challenge. So far, the field, before I was trying to work on this problem, has relied on relatively mechanical ways of doing it. For example, we start with a pre-validated building block, and we have geometric sampling I just talked about, and then they dot architecture. These structures are highly porous, and then they rely on very large interface. It's nothing look like on the left side, where this naturally selection-based structures. How do we change that? This is what I want to propose, which we worked on, is a top-down approach, where instead of using a pre-validated building blocks, we want to specify a end product that, for example, have some geometric constraint. For example, this size, making proteins that fill in these excluded volumes and then having low pores. And then we can utilize some AI mechanism or algorithms to allow them to design custom building blocks to fill in these strategies. These are architectures. How do we do that? Well, I play a lot of computer games as well as board games before, so I got some really cool inspiration from the reinforcement learning algorithm. For people who don't know about reinforcement learning, it's an algorithm that or a set of learning mechanism were being very prevalently used in games, board games. For example, in 2016, AI defeat top human players in board game Go. Board game Go is thought to be the hardest game people have invented so far, and then AI was never be able to beat top human players. But that was proven wrong in 2016. The mechanism behind the the algorithm that beat top human is so-called multicolored tree search, which the mechanism behind that is you can make moves constrained by game rules to maximum reward or win the game. So they train scientists from the DeepMind trained the program based on these searching mechanism. As you can see, for example, in the random time, and then you are able to make your moves by placing a black piece. You can have multiple options, and then you can calculate the value of each move and then try to figure out what is the best move to make. That maximize the expected total reward, not the immediate reward. So that if you just take the top reward of each single step, you may not be able to reach the global minimum or the best maximum. But you can do that by a lot of roll and play to choose a move and then create a new state and then do many, many steps until you win the game or lose the game. By winning or losing, you're able to kind of punish or reward each steps that allows you to make these search tree. So after that, you can train a value network and policy network to actually help you to guide through these process to make decisions so that you can make better decisions next time. So it's an iterative learning process. And what more impressively in 2017 was people no longer need to utilize the value network and policy network trained on top human player moves, such that you can just directly do self-play and then improve, and that will be able to beat the previous generation. So my hypothesis, can we take advantage of what people have used to create the Go game to create to design proteins? That's just basically replacing what our job as a protein designer, so that the machines can start to design by making an AI agent that make reasonable moves and then design better protein. So the idea becomes, how can we grow protein backbone to maximize predefined scores? For example, fill in the target volume, foldable backbones, or make good interface. Each can be assigned a specific value. So what we decided to do was to orient assemble protein secondary structure. So it's more like a fragment assembly approach, because we don't want to have a lot of different moves. So we picked a relatively simple move. It's basically growing the helices and then making a turn. So basically it follows growing and helices and make a turn. And then we can check for clashes with neighbors. In this case, I'm showing a icosahedral capsid just because it's aesthetically pleasing, as well as a more complex system. So that instead of just building one monomer, you can also building all these complementary partners at the same time by applying asymmetric matrices. And finally, you can just create a score term where all these monomer clashes, you get a huge penalty, as well as if you have making a good core, you create a good, you're rewarded. So basically, instead of trying to understand how proteins to fold, we can make the game rules and then let the AI agent to try and error and then start to design proteins. And it actually works. So what we turned out to do was, as you can see previously, making the actions where the monomers can grow sequentially by fitting together secondary structure. Well, so far we are just showing the secondary structure of helices, but eventually when we improve the algorithm, you're able to making strands or other things, and then symmetrically they're making icosahedral. So during this rollout stage, you can start with a random rigid body initialization, and then you start to making exploration by expansion. And then whenever it gets a score, it will immediately update the search tree such that the weights are updated. So the next time is not, initially all those options are wholly random, and then you start, when it's making iterations, it start to make better moves and better moves. And eventually, if you just wait until it's finished the entire search, you can also get a final evaluation based on a lot of score functions, and that allows you to back propagate the weight of individual search tree. So that's a mechanism behind it, but we have a lot of things we can discuss in detail. Here is a training process. As you can see, it's just you do the random rollouts. Initially, without training, the performance or the scores we assign are relatively bad. And then the protein designs are relatively, for example, the tight packing scores, these proteins are not making a tight compact core, but it eventually gets more compact area, structures. Also, this interface designability previously is not making a very complementary interface, but now after training, it gets to make very complementary interfaces. So that allows you to make really compact structures. Other conditional generation of structures we can also do, and that can be conditioned on the volumetric constraint, so that, as Corey already mentioned, we have a lot of engineering. We want to push the biological engineering to more traditional engineering approach, so that we can blueprinting parts. So that's how we can do it with this strategy. We can just making a blueprint of volumetric input and then making protein that fit in that particular shape. And then also these design pipelines are fully automated. You can just do one click that starts from a backbone of the monomer. And then for these bottom details are for expert. For general audience, we're just focusing on the things in the box. We can have a backbone generation that one click you get you these assemblies as well, the monomers. And then these monomers can be designed through different software, for example, Protein MPNN or Rosetta. And then you can down select by applying certain filters to design the interface, and then iteratively resample around this close proximity, and then finalize, diversify the sequence. So one click protein design possible, and then you can get your own capsid. So does it work? It actually does. So we tested around 100 designs. We have a relatively low success rate for this particular task because we didn't quite optimize things throughout the process, as well as this test is kind of challenging. So we were able to create something similar to a ferritin protein that's 12 nanometer, composed of homosextimer. CD spectra suggests these things are highly stable, as well as the monodispersed inside the inclusion chromatography. We also performed a cryo-EM to characterize the structures. As we can see over here, the gray is design model. The color is experimental data we obtained. They almost match perfectly with respect to the monomers in both cases. And then the RMSD stands how much deviation from your design structure. The deviation is less than angstrom. It's like less the angle of the distance of a carbon-carbon bond. And then in this case, the RMSD of the entire capsid is only 1.4 angstrom deviate. And all the interfaces we are seeing over here are closely resembling what's our design model. So it's just like a test of case where we see that some of the most challenging tasks can be designed through this strategy. We also can create a lot of robust fusions onto these proteins because they're so tiny. Homosextimers of around of 60 residues. They resemble one of the smallest not one of the smallest icosahedral structures. We can utilize them to display molecules at a very high density, as well as them to attack a nucleus localization sequence so that they can get into cells. In this case, we attached a multivalent type 2 receptor superagonist so that these Ang1 protein were able to facilitate wound healing by appending them to this homosextimer. They'll be very compact superagonist so that we can see that the signal is much higher than all the existing structure we will be able to see, and then twice as high as the same valency of this larger particle, just because they're smaller and more compact, so they can signal better. We also be able to utilize them for a influenza vaccine by displaying this influenza virus hemagglutinin molecules on the terminus. So as you can see, by displaying these stems of the HA, you make these protein subunit vaccines. And by comparing them with the state-of-the-art protein subunit vaccines in the clinical trial already, we see that's a higher titer of antibody generation from these architectures, just because we rationalize that potentially due to their compactness. Lastly, as a kind of example, we can also make these nanopores based on this strategy, where we can see a reading pore and then this host ring that allows them to be mobilized in the silicon, so that the cryo-EM structure confirm these structures can form, and then we can mobilize them into chips for a bioelectronic applications. And lastly, just one more small side project. We're able to make small molecule binding proteins, and then in this case, a chlorophyll binding protein. Its chlorophyll dimers are designed into this protein pocket, and then we make them into a cage so that it's a supramolecular light harvesting as well as a reactive center protein, and that you can do with high valency. So potentially we can look at the photosystem synthesis side of things, these architectures. So in summary, I would like to kind of share with you today, the take-home message is computational protein design is an emerging tool that people in the synthetic biology field should think about and including in their pipeline. So we heard a lot of great things from Corey about programming the signal transduction and how things communication. However, we are able to innovate some of the function modules that primarily rely on natural proteins. Here, we can design powerful proteins together, and then we can replace some of that modules. That creates a lot of opportunities out there. And then what I'm showing you over here today is there are increasingly powerful algorithms out there. This field has been open to all the computer scientists and then biophysicists, and then things will just get better. And second of all, this turnover from computers to cell biological systems are very fast within weeks or so. So we should really think about how we can engineer a biological system in the next generation. How do we do things? Here is a vision I would like to share. This is the kind of time and complexity of protein design systems where we can start from the monomers or assemblies, enzymes, or certain simple enzymes, and then small molecules, nucleic acid and peptide binding protein. And Corey mentioned about transcriptional factors. This is what we can do right now. What we haven't been able to do, some of the supramolecular system, is can we make carriers of, in this case, like viruses for delivery or smart therapeutics, or machineries that complex, making very difficult functional tasks, or interactome that deal with the fundamental biological system. So a sustainable future will probably rely on some of these advances that we are seeing for us for the future. So with that, I would like to kind of acknowledge all my academic mentors and collaborators, as well as my PhD advisors. And thank you very much for your attention.
Thank you so much. This is an amazing talk. Again, I now remember your amazing presentation during the conference I met you for the first time. And also, I again thank you for your great explanation about reinforcement learning. That is absolutely amazing, especially using the Go game. Actually, as I mentioned earlier, I play Go game very well. So I also I cannot help thinking about or saying this one now. I hope reinforcement learning is more quickly applicable to or for raising my teenage daughter than training AI. I right now feel like mentoring, advising, raising my teenage daughter takes much longer time than training AI. Because you just simply publish one paper in Science, I believe, within a few years or a couple of years of effort. It takes probably 50 years for me to train my daughter in somehow. Anyway, that's my joke. Well, so one question I want to ask before I didn't have any question for now. The question I have is, I cannot help thinking. Actually, the last 2 days I watched one TV show called Hidden Singer. The Hidden Singer is basically a TV show. Multiple guy compete each other to impersonate the main star's singer. And then the difference between star and the impersonator is the person is who are star is basically the creative or creator of the art form, and the other competitor is basically copying and and then try to mimic. So to me, at this moment, AI, all the, AlphaFold, and then the program you developed, Dave Baker already kind of show the de novo design of the DNA. And I believe that's his creation. But still, many things kind of rely on the previous data. There's the crystal structure, a lot of data in database, etc. So to me, that is still like impersonating somehow the old database, old learning, old knowledges. The real question to you is, do you foresee in near future we really generate AI or whatever algorithm that AI really do create things from scratch? I mean, I'm not talking about de novo design of the protein. I'm talking about real function design. We already seen some initial stage, but more prevalent, more kind of easily. Even me could do something by using the program to create something new. That's dangerous in some sense, but still exciting. So I want to hear from you.
Wow, that's a very provocative
Of course.
question. I feel like that's very inspiring. I try to answer that, but with my limited knowledge. So I want to kind of first make a clarification. The algorithm I did, the reinforcement learning is a kind of interesting scenario where it doesn't rely a lot of data. So I just want to briefly explain. This typical deep learning is trying to learn a probability distribution of data. For example, making a cat. So you take a lot of cat images and then try to copy that. Or it's like generative model is trying to learn from previous existing structures. The protein, most of this state of art protein language model or protein generative model are relying on PDB structures. So they're making proteins that's similar to the natural protein. There were structures that have been determined as structure. Proteins has been determined structure. But there's a bias, right? Things already exist or things can be solved with structure. Well, the nature does or the evolution does was trial and error. That's just to go expand, randomly do the applied selection pressure. So that's how it creates new things. What the reinforcement learning is, in our case, as you probably already learned a little bit about, I didn't train on any particular data set. I didn't mention that, just because we rely on trial and error. So it's reminiscent to a certain extent more of how nature does things, but it accelerate that in a chip. So that brings the philosophical question. One is you can either try to mimic, as you just mentioned, what nature does, or you can do an accelerated evolution on chip, in silico evolution, by doing the trial and error. So that becomes a sampling problem. The other one would be just a mimic problem. So I think in the future, if you want to truly create something, you want to expedite the sampling process and design your system that so basically create a force field that resembling these natural selection or biologicals. So instead of trying to replicate what's the structure look like, try to create an environment that looks more like a natural environment, creating the force field or functions, and then do the selection in silico that way. That's essentially how we did it for the reinforcement. The data didn't present as a training model of what it should look like, but the score function, how do we assign scores? How do we score the post? That rely on some empirical knowledge, which came from data indirectly. But I just, this is kind of my shallow understanding what's possible to generate truly innovative system is instead of trying to replicate what's existing over there, but to create a big simulation box that looks like the environment you want to behave or the selection pressure.
So that's actually excellent answer, and then thank you for the clarification, because there should be big clear distinction based on the machine learning or whatever data based kind of thing, and also the reinforced whatever learning, that's a little bit different. I realize, also remember the clear distinction. No, actually, the question I originally want to ask, think about this way. So based on my narrow knowledge about Go in AlphaGo, for example, AlphaGo play and try multiple different, actually, whatever countless, combination of thing, and then thinking about how to win the game, right? So that means AlphaGo thinking the goal is the winning the game, and then try multiple different weird move or whatever trial and error, but they only choose the best solution to win the game.
Yeah.
In contrast, the thing I want to talk about is I'm talking about creation or creativity. In contrast, my daughter or teenager in general, or whoever creative, the solution they choose obviously not the best solution for at any given time. But sometimes that lead to real breakthrough, innovation, creation, something completely unexpected. That is the difference between, I believe, AI and current AI, I would say current AI and the human being. And that's how human still unique compared to the AI. One day some crazy guy may make AI also feel, behave like my teenage daughter, unexpected way. That is a sometimes disaster, often disaster, I would say. But sometimes that kind of make the breakthrough innovation. So that actually my question originally wanted to ask to you. And then do you have some more insight about
Well, I
that?
wouldn't say insight, but I think that's a very good story. It's getting a little bit more philosophical than biological, but
Sure.
I'd like to share a little bit is, yeah, so that's humanity. It's a kind of random walk, and it's more like the process because the machine will probably just do gradient descent. It's just the math that takes probably the best way of getting to your goal, and that's so predictable. You will just get the best you can get is to mimic your data in certain ways. However, in human, we make mistakes, and we have serendipitous discovery that be very delightful and then make our day. Your daughter or everybody's imperfect, they make mistakes, but they're beautiful mistakes. They take turns. They don't, for example, they turn out to, I'm not saying, but maybe she turned out to be a singer, but all her college education instead of like art education are potentially useless. However, that will make her has like find a great partner or like have other taste of things. Just the life is so unpredictability of
Sure.
life makes
Sure.
it exciting.
Yeah.
I mean, yeah,
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 so this is the probably takes another 30 hours to discuss, but my point is sometimes my daughter is predictable because she disagree with me anything about some topic, just intentionally disagree with me. That's a teenager, but that's a predictable. But the other thing is not predictable at all. So, we can talk about that later. But absolutely, wonderful answer, wonderful talk. I really appreciate it. Let me check whether we have question from audience.
no,
Uh,
I don't.
I don't have see anything, but we also now 9 minutes after 11, so let's close and then we chat afterward. So thank
Okay.
you so much. This is absolutely amazing talk. I really delighted to have
Well,
you.
thank you so much for having me.
Yeah. Also, Corey need to go to another meeting, and he said, this is a brilliant, amazing talk, in the chat. So I want to deliver that message to you. So to close, thank you all for joining us and staying with us today. We'll meet again June 6th, Thursday, the same time, the same Zoom link. I will be joining you from Korea at midnight local time, unfortunately. We'll have the Professor Jacqueline Niles at MIT and Dr. Jeremy Zhao at NYU, the New York University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us, and I will promote you to panelists who can speak and show your handsome and pretty faces if you wish, without recording. Thanks. I will stop record. Just 1 second.